Ladies and gentlemen, may I have your attention, please? Welcome to the first talk in theory, but the second talk in theory, but the first talk in practice of this session. The speaker is Professor Fengzhu Sun from University of South California. He is an expert in informatics and is going to talk on statistical and computational approaches for investigating virus host interactions using metagenomic high-C data. Now let's welcome the speaker.
First, thanks for attending my talk, and also thanks the organizer for inviting me to give the talk, and for Dr. Li for the introduction. Last year, I gave a similar talk on virus host interactions, mainly use sequence data only, with a brief touch on metagenomic high-C data. And this is a continuation of that talk, and in this talk, I will concentrate on metagenomic high-C. To bring us to all the same background, I would like to first introduce some metagenomics and the metagenomic high-C, and then we talk about our recent method for analyze metagenomic high-C data, and that's including normalization using high-C and meta-CC, and then we developed several computational methods for the downstream analysis, including high-C for binning of those microbial genomes. And then we also developed methods for identify viruses from metagenomic high-C data. That's the viral CC. Finally, I will give some brief summary. So metagenomics is the study of genetic materials from the environment, such as from the ocean, from the soil, or human bodies parts, such as the human gut and the skin and nose, etc. So because of those wide applications, there have been many large-scale metagenomic projects like the Global Ocean Service, the Human Microbiome Project, the Metagenomic project from Europe, and there was also the Earth Microbiome Project and the National Microbiome Initiative. That's including all the different environments, such as from the ocean, soil, and the human body. For all those large-scale projects, and mostly we concentrate on bacteria and archaea, with little attention to those mobile genetic elements like phages and plasmids and other type of mobile genetic elements. Recently, US NIH started the Human Virome Project, and the objective of the Human Virome Project is to identify all the different viruses, including those zoonotic viruses, and as well as phages that affect the bacteria and in turn affect the human health. Here is some simple data from the water, because my collaborators are mostly marine biologists, and so we have a large number of viruses, and so you can see from this data that the viruses is about 10-fold in terms of number, is 10-fold higher than the number of bacteria and archaea. Then viruses, although in those phages and they affect bacteria, and we all know that viruses can cause a lot of changes, impact the environment, such as the SARS virus, that's the SARS virus, COVID, and they just have an important impact in the society. Then, I just mentioned that most studies concentrate on the prokaryotes with little attention to mobile genetic elements. Then, what type of questions we are interested in those viral projects? We are interested in what kind of viruses are there and what their abundance and how the viruses impact those their host, like virus, their host, and in turn return the host, like the human body or the ocean or soil, etc. Then so the scientific question we want to recover the novel microbes. In particular, we want to recover novel viruses. We identify those active viral-host interactions, and we also try to track horizontal gene transfer in the microbial community. To study this kind of problem, there are different methods to do this, and so one particular one is the use sequence data only. And in particular, for the last 5 or 6 years, we have developed a large number of machine learning based methods to identify viruses and like viral finder and the deep viral finder and the virus host matrix, etc. We developed many methods that can be used to identify viruses in the microbial community. And that was the topic of my talk last year. And so for this year, we are going to talk about metagenomic high-C. And so for metagenomic high-C, we have 2 types of data. The first type of data is the so-called short-read data. So this is the example that we have. I mean, suppose in the community, we have a variety of different microbes. And for those longer lines that indicate those relatively large microbes like bacteria, archaea, eukarya, etc. And those different colors indicate the different type of microbes. And then in the environment, in addition to these relatively large microbes, there are also small microbes like the phages. And here we indicate those different type of phages and the plasmid and etc. So they are much shorter than those bacteria. But that's all we know, but we really don't know what they are. To study them, we use the so-called next generation sequencing to sequence those genomes, and then we get so-called the short read. And so we have many of these read, and then usually in this some short read data we have around 200 base pairs or 400 base pairs for both end, and then we get many of these short read. That's where the computational biologists come from, based on this type of data. So there are many computational problems to analyze such kind of data. The first type of computational problem is to assemble them. Because we have the short read around 200 or 400 pairs, we want to extend them. And so in that sense, it's how do we connect them? So in the simplest idea is to say if 2 short read have some overlap. If they have overlaps, we connect them, and then we get relatively longer compute. And so there are more advanced computational methods to assemble genomes, but here we are using other existing tools. We are not developing computational method to analyze, to assemble this. Once we assemble them, we get relatively longer regions, genomic sequences. We call them the so-called contigs. And so contigs are genomic regions that are continuous along the genome, and it's much longer than the short read data. Some of them can be thousand base pairs, some of them can be tens of thousands of base pairs. So that's the first. So for our work, we concentrate on here, and then we have the context. We ask, okay, so for this context, all these we have are the ACGT. And so we have the sequences, and we don't know anything about it. So yeah, simple way to do that is to try to map those contexts into the reference databases like the ACPI database, and then you see what they map to. And if they map to some known genomes, and then you know where they are come from. But the problem is that for many of those read, probably half of them, you cannot map to those reference genomes. And then in that sense, are they come from bacteria, archaea, or viruses, and so on. And so in that sense, if we cannot map this read to the reference genomes, we want to classify them. And then that's getting to the so-called machine learning things. And because we already know a lot of those sequences from the databases like bacteria, archaea, and the phages, and so on. And then we can use them to learn what are those sequences from. And so using those training data, and then we just classify them. So again, those are the methods that we developed for many, many years, and that have a big impact on the community. And like ViruFinder, Deep ViruFinder was used to build new phage databases that is quite useful to those national virome projects. And so that's the first type of data that we have the concept of the read, and then we have the context. What I would like you to remember here, concentrate on is the context. It's just longer regions, and it's not the whole genome, but it's a longer context. Okay, so that's the first concept. So the second type of data is so-called metagenomic high-C data. So now we have those contexts, but we don't know which contexts are from the same genome. We want to group them if they are from the same genome, and then we want to group them together, and so that we can get the new microbes. Okay, so that's the idea. How do we do that experimentally? And say those is one method that can be used to see whether they are from the same cell or not. And what's the idea for those experiments? Actually, it's not new. It had been used in like human Drosophila in single organisms. And then they study the relationship between the different parts of those human genome, try to see which part of the human genome are physically close to which part of the human genome. And then they can study gene regulation. And this metagenomic high-C is using similar kind of idea, but it's used in the metagenomic community. Okay, so the idea is the experiment looks like this. So first we use some restriction, some biological enzyme to link those genomic regions that are physically close together. Okay, if they are physically close together, they are more likely together. Okay, so now you can see here, this is one part of one genome, and this is probably is from a different genome if they are in the same cell, and then they are linked together. Okay, so we first link them. Once we link them, we use some restriction enzyme to cut it. And so you can see here is we use restriction enzyme to cut that, and then some of them might be linked, some of them not linked. For those ones not linked, we wash them away. We wash them. And then we get the so-called linked regions. And then we use ligase to ligate them. And then we have the ligate sequence, and then we sequence those parts. We sequence this part. And we sequence them, and then we get the hybrid read. And the one part of the read come from one part of the genome, and the other part of the read come from the other genome. Okay, so we have the hybrid read, and then we use this information to see which genomic reading close to which genomic reading. Okay, so in the metagenomic high-C data, if they are from the same cell, they are more likely to be linked together. So that's the idea. From there, we try to group those contigs and try to say, okay, so which the genomic reading link together or which contigs link together. Okay, so that's the basic idea. Any question for the data? Okay, so it's good. So the next step is the same, conceptually simple, and then let's see what the problems are. So conceptually, actually, what they do is the following. So here you have the shotgun read data, and then we do the assembly, we get the contigs. Okay, we get the contigs. And then we have the other type of data, that's the high-C read. And remember, the high-C read are hybrid. And one part from one part, like one end comes from the one part of the contig, the other end comes from the other part of the contig. And then we just map them. And then this is one contig, this is another contig, and then this is the high-C read, and one part linked to this contig, and the other part linked to this contig. And then we see there is a high-C link between this pair of contigs. And so conceptually, we say if they are in the same cell, and more likely they will be grouped, they have more links. Okay? And then, yeah, we have from here, we get to mathematics. And now we construct the graph, and the node indicates the contigs, the edges indicate the number of high-C links between this pair of contigs. Now you have a graph. From this graph, and then we do graph clustering. And then all you learn from graph clustering, you can cluster them, and then that's most likely those ones that are grouped together, they are from the same genome. That's the very basic idea. And so given some features, affect some hosts, and then because they are in the same cell, then you see there are some viral host interaction. That's the basic idea. But the problem is there might be some kind of bias. And remember, we are dealing with real data, not some conceptual. And so in that sense, all the experiments have some biases. And are there some biases for this metagenomic high-C data? So now we try to see if there are some biases. So to do that, I would say is we explore the data. We say, okay, so this is from the real data, from the real experiment, and we know what kind of cells we put in, and then we try to see whether there are some links between them. And we go through this procedure, and then we just see the relationship between the number of high-C links and the several potential factors that impact those high number of high-C links. Okay, so now this y-axis is the number of high-C links between a pair of contigs. Okay, so each node indicates a pair of contigs. And this y is the number of high-C links between them, and the x-axis, and depend on what like this this figure indicates the product of the number of sites for these two contigs, for these two contigs. Okay, so because it's the number of restriction site, and then the other one is more easy to understand. That's the length. I mean, we have the contig length, and then for the last one is the so-called abundance, Hall abundance. Okay, after you explore this, and then you see a clear positive relationship between those number of high-C links with the number of restriction sites, the length of those contexts, as well as the abundance of these contexts, right? Okay, so what's the problem? The problem is our assumption is if they have more links, they are more likely to be in the same genome, right? But if you use some threshold, you cut the threshold if their number of high-C links is greater than this threshold, they claim that they are from the same genome. The problem is the longer contexts tend to be called they are linked, right? They are from the same genome, or more abundant ones from the same genome, right? And this way, but then you ignore those short ones. You're less likely you call those short ones to be linked together, although they might come from the same. So there is a bias there. And without correcting this bias, and then you will have lots of false positives and lots of false negatives. And then you will get the wrong result. Okay, so then that's just the first step we need to do is normalize the data so that the number of high-C links will not be associated with these factors. Okay, so in this case, we borrow the idea of normalization from other type of studies like gene expression analysis and so on. And so then we just use the so-called the zero enriched negative binomial distribution. Okay, and this has become more mathematical, and that's why I'm giving talk in the mathematics conference. And so that's the so-called zero enriched negative binomial distribution. So now the number of high-C links, that's the Zij, is modeled by enriched zero enriched negative binomial. And so we have a probability of pi ij that is just no linked. Okay, it's zero enriched. And the other one is it's given is not zero, and actually it's a negative binomial can also have a zero. And then it's another component is a negative binomial distribution. And so with the probability 1 minus pi ij. And so there are two parameters, is pi ij and uij. And from here we know that those pi ij and the uij all depend on the number of restriction sites, the length, and the abundance. Okay, and we want to remove those factors. And for this case, what we have is after we do some mathematical analysis, what we get is here. And so like the expectation is bound on this, and then we do those fit those model using our real data. And in this case, after we estimate those parameters, the pi and the beta, and then we can calculate for any pair of sequences, context, we can calculate the expected count of the number of high-C links. And then we remove them. How do we remove them? We divide them. And we rescale them using here the Eij is equal to the observed count divided by the expected count. And this actually is just similar to other type of normalization method. Those type of model actually you can see it's not novel because in the sense it had been used in many other gene expression analysis, other next generation sequencing type of data. On the other hand, this is the first time that those negative binomial distribution was used for the metagenomic high-C data analysis. Okay. And so after we do this normalization, we try to say, okay, do we remove those biases? And so now essentially we just, again, we try to look at the association of the count with those factors. And so without any normalization, and then you can see there are high association between those number of high-C count with the number of restriction site, with the length, and with the copy. Okay, And so if you just do like the number of sites, the normalization, and then you cannot recover those coverage. And similarly, those length, simple things will not work. And this is another normalization from the other groups. And then you can see they are doing a decent job. And then from the naive negative binomial, and then yeah, it's okay, but still you are not doing well with just remove the bias for the coverage. After you use the negative zero inflated negative binomial, that's our method, and you can see that those correlation coefficients become much smaller. So that's the first work that we started on this subject, that it was accepted by the RECOM. Actually, this is a simple idea. I first proposed this idea, and my students Yu Xuan Du, and they finished this in one semester and get accepted for RECOM, one of the top conferences in the field of computational biology. Okay, so that's everything sounds good. And one problem that I raised to him is the following. Nowadays, with the technology improved, and then we get people are using long read instead of short. I asked the question: is can we use our normalization method to those long read data? And then he tried, and we hoped it worked, but it didn't. Then what are the problems? So there are two problems. And the first problem is the, yes, yeah, this way. Oh, okay. So yeah, thank you. You can think it's the same thing. Yeah. Thank you. Huh? The IJ is the pair of contigs. One contig is I, one contig is J. Okay, so yeah, it's the okay. Then yeah, we try to apply this method to the long read data and also to large scale data. And then uh, when we find the method, although it sounds good and it's for small data set it works well, but when we get to large data set and the computational speed and also the storage get into problem. And so the one problem is that this will not work well. One is the problem is the computational complexity. We are doing a pair of contigs. And so let n be the number of contigs, and the computational time will be O n squared, right? And yeah, you already heard a lot about the computational complexity, and so once you get to n squared, when you get to the large data set, then it will not work. Okay, so now how do we analyze such of data without those square? Instead, can you get some method using linear computational time? So that's problem one. Problem two is the so-called CI. And we try to estimate the coverage of the config using high C read, using long read. The problem with long read is that they have relative, the number is long, and then they don't want to sequence a lot because it costs money. And so the number for high C read is small. Okay, the number of high C read is small. In that sense, you cannot accurately estimate the coverage just because the number read is small. So in that sense, you cannot do that, and then that's the method will not work for the long read data. And can we improve our method to normalize those long reads? Okay, so that's getting to our next project. It's that's published in Nature Communications, and so okay, so the idea to solve the first problem is to reduce the to reduce the computational time from n squared to n. Now, instead of doing pair of counting, we try to model the number of high C rate mapped to one particular counting. So, for example, this ice counting, and we just look at how many high C rate that are mapped to this one that links to the other. And so we just sum over all them, and then we model this one using the zero enriched negative binomial. Okay. So in this case, and because now we are modeling individual contigs instead of pair of contigs, and then you get the linear time. So that's the first. And so the next question is, how do you overcome the problem of coverage? As I mentioned, the coverage cannot be accurately estimated for long read. And so we don't want to use that number actually. And in that case, and how do we do it? And what we do is using the so-called the high C count, the high C read to map the to calculate the coverage instead of the like the using the contig to calculate the coverage. And so here is we try to look at the number of high C read that map to the same contig. Because remember here we have longer contigs, very long contigs. And then there are a lot of read, high C read mapped to the same contig. And so now we have this one, and then we again we model it by the negative binomial distribution, and then we do some mathematical manipulation, and finally it what uh, we get is I mean I due to time constraint I'm not going to give you details, and for those of you who are interested, you can read that paper, and then after all this mathematics, what we get is yeah after all this mathematics, not really that advanced, but it's need some mathematical calculation. And then we just model this UI by now still this is the number of sites, this is the length of the county, and this is the NI, that's the number of high C rate mapped to that county. Okay, and this actually is reasonable because now we just use although we don't use the read the regular read, but now we are using high C read to calculate the and then we get this. Okay, with all this, we solve this problem, and then the next question is, now we are not modeling the read pairs, but we kind of the each individual, and now with those the number of high C count between these two contigs, how do you normalize that? We use this to normalize. We just have the UI estimate UI and the UG. We just use this one to the expectation. We just assume that these two are independent. Okay, and this thing it actually is not really relevant, just kind of for computational rounding errors. Okay, so now with all this, we can get this normalization, and again we kind of see, did we solve this problem? And so one is computational time, and the other one is do you get the good downstream result? Okay. So now with this one, and so do we get remove those biases here? And so for this one, we can see this correlation between this high normalized high C and the number size and the length and the coverage. And for this one, although it's slightly larger than what we did before, but it's I mean you can still see here the correlation is effectively removed the biases. Okay, so that's the one. And we remove that. And so the other question is, did we speed up? So now you can see here the computational time. And so these are the real microbiome data. And then we can see here is roughly you can this is kind of even for the before when we have a lot of setup time, but this if you just compare this computational time, and then I counted it's roughly around 60-fold decrease in terms of computational time. And that's we also get some quite decent result for the downstream analysis. So yes. This what the hours means. The, so for the first method, we need to do some kind of pre-processing, and that's pre-processing time. And these are the real computational time. And this one does not need even pre-processing. Okay, so now we have this normalization method. And sure, I mean, normalization method is just the first step. And then the next step is, can we get novel biology out of this? And that's the next question for the downstream analysis. So for downstream analysis, we first try to recover the so-called metagenome assembled genome. So essentially what it means is, remember here we have the data, we have the down data, and then we do the normalization. Now we remove those biases and we remove those spurious rates, and then we can get the so-called normalized graph. We can get a normalized. And hopefully, with this normalized graph, we can get more biology out of it. Okay, so as I mentioned, the objective of C is to recover the microbe genomes and study virus-host interactions. Can we do that? Okay, so that's the next phase. It's kind of getting deep into biology. And so now this is the step. I mean, you already see similar figure before. And so here is the data, and then we get the raw count map. And then from here, we add those steps. And so we add this one using the our normalization method. And so because the first paper is we do the high CC, and now we can use those meta CC, and then we also remove those spurious interspecies contact. And then after this one, you can see all this is just changed here. And then we do here, and then we use the so-called leading graph class, and then we classify those contexts into different clusters. And then we have this, and then we ask, will these clusters close map to the real genome? And that's the next question. So this is the computational method, and then we see whether we get it right or not. Okay, so now we use real data, and so one is the fecal sample from a human subject, and also we have water samples. And then we try to, once we have those things, we try to ask how complete those genomes are, and are they contaminated or not. And so this is the standard algorithm, that's the check arm, other people developed to see whether the genome that you recovered are complete and how contaminated. And in the community, there are some standard criteria to see how good your cluster is. And then this included the so-called near complete genome. So if you have a clustering, and then you just say, okay, so now how complete they are and how contaminated they are. Okay, and then we call this as the near complete. That's the 90% complete, and with the 10% contamination, and the substantially complete, and that's 70 to 90 completeness, and the 10% and moderate. So yeah, these are just community defined criteria. And then we compared our method with several other methods to for this type of problem. And so here is the for this one, and then this is for the human gut data set, and then the Y is the number of clusters or number of metagenome assembled genomes, and then all different numbers. And like this one, this column is the number of max that are near complete. And so you can see here, this is our result. This is the IC bin, and so you can see here we recover around 70 complete genomes, while the next best one, that's the Metatol or the B3C, then they the B3C, and then they only recover 60 complete genomes. And if you look at the all the total number of recovered max, and then we recovered much more than this second best. And similarly for this wastewater data set, you can, I mean, although this total is not, but it's the most important one is this one, the near complete. You can see we have close to 100 recovered near complete genomes, but it's the other ones they don't. The next best is just 40, around 40 complete genomes. So yeah, you can see this after normalization, you can get much better results. Okay, and that's one application. And the other application is, can we recover viral genomes? Because we are interested in the viruses. And then if we just use that method, what I just mentioned, you really don't see a lot of viruses. Why? And so that's the question I raised to my students. And then he found the following argument. It's, remember, for viruses, it's short. Okay? And it's short, and then, sure, then because it's short, and then the number of viral contigs will be small. And then if you try to find those links, it becomes quite challenging. The graph is very, very sparse for viral contigs. The sparse is very sparse, and then you don't get the complete viral genome. And then that means we cannot use our previous method to get that. So what other information we might be able to use to get the viruses? So we just try to enrich this this graph so that it becomes more dense. So he used the following idea that is quite novel, and at first we thought it's kind of controversial. And so he used the following idea that if two viral contigs are from the same virus, and then they are more likely to link to the same host contig. So he tried to use those host contig information. So although these two contigs, you cannot connect them because just based on the Hi-C link. But if they are linked to the same contig, host contig, and then they might come from the same gene. And that's the quite a novel idea. And at first we have some doubt about this assumption, but he worked very hard to show that this is the, uh, the right way to do it, and it works. So, then yeah, here is if you understand what I just talked about, is he just used those host contigs as a mediator and try to link those viral contigs, and then that's he integrate those contigs, the graph from the direct linkage plus those host mediated contig graph, and then get a unified graph, and then he do the graph clustering again. So yeah, this one you can see here, and again it's the same thing. And so we just try to identify those viral contigs that we use our program that we developed before, and we have the high C rate. And then you can see here, the first step is just the construct the high C interaction graph. This is just before, and then we also construct, this is new, and we construct host proximity graph. That's just the host mediated graph. And then we integrate those two graphs, and then we get the unified integrated graph. And then we do the latent cluster again, and then you get the viral genomes, and then you try to see whether there are some linkage between this virus and some host things. You use high-C link, try to see if this host genome are linked to some bacteria genome. And so then you can study the viral host interaction. Okay, so then what we get, yeah, this is just the methodology, and just using Word. Then again here you just try to see those mock communities, whether we cluster things right and so on. And what we have is just again is because we know the clustering of the viruses, and then we can calculate the F score. And so essentially what we just try to say is, yeah, we get the good result. And for this one, we also try to see how many viral beans we can get. And so this again is similar to like before. We have the so-called near complete, and then we get much more near complete viral genomes than the other method available. And so yeah, you can see it's a similar figure here. And for this one, these are the real data. And then we get how many of them are just complete, and so these are 90% complete, and then the different colors get used, and this one like 80%. And then this is our method. You can see here, for this real data, we can get more complete viral contigs than the other method available. Okay. Okay, so for viral genomes, and then we don't have a criteria for the contamination evaluation, but we try to develop another method to see whether the complete genome, the genomes we constructed are contaminated or not. And so for this one, we see for one cluster, we get all the protein sequences, and then we map it to the protein sequences, the database, and see whether they come from the same family or not. And then it turned out that they come from about 90 or 85% of these retrieved viral genomes, and the proteins that they come from the same protein family, and from the same family. So indicating that what we get are also pure. Okay, and so that's the interesting result, and we show that what we get are pure, and also we show that we get more viral genomes than the other programs. Okay, so now we study getting to the core is the link, the viruses, and the host. And so here is we can see this is just for one particular viruses that the in community. I forgot. I suppose this is from the ocean. And so you can just draw this figure, try to say, okay, so what are the abundance of different viruses, and then you can see how abundant they are, etc. And this just give you the abundance of the different viruses. And then in addition, because we can link those viruses to their host, and then we can study, okay, so a particular host, and then what type of viruses that might impact those particular hosts. And so in particular for this example, we can have this particular host family, and then we look at all the kind of viruses that we find that are can affect those hosts, and then you can get a distribution here. So now with this method, we are able to calculate those the host virus interactions. Okay. So with this, all this kind of method, we also developed some other method using sequence-only information to do clustering. I'm not going to give details about this one, but we try to include both sequence information as well as high-C information to link to clustering the context. And finally, I would say that Yishan, my student, is I mean, he's great. I mean, he just built, in addition to this, all these different good publications, and then he also built a very useful, very user-friendly software package that just published recently on NAR. And for this package, he just combined all this methodology into a user-friendly package. And now you can see here that the normalization value and using those high-C then or the meta high-C, and then just remove those various high-C contacts, and then they do the clustering and the visualization of the different context, and then they study the different like viral host interactions. And so this is just he's a great student, and so after 5 years of study, and he did all this the methodology development and the software, and actually he is my first student who directly get a faculty job in the computer science department, and that's quite rare in our community. So so in conclusion, we have the developed normalization method for normalize the high-C data and detect various interspecies contact, and then we do those normal, I mean, the downstream analysis, high-CP, for assemble those the bacteria, archaea genomes, and for virus CC, that recover those viral genomes. And then we are continue these studies to viral host interactions. After this work, and these are kind of, we get some other collaborators getting to us and try to collaborate with us on like the viral host interactions in different environments and so on. And that's what we are doing these days. So finally, I would like to thank my former students, and so Yishan Du, and most of the work that I talk about are done by him. And so for the work that I talked about last year, they have a lot of students involved for the study the viral host interaction. And we also have many collaborations with the marine biologists in particular. I would say Jed Furman helped me greatly in terms of identify those kind of scientific problems and apply this kind of tools to their studies and so on. And this is supported by NIH and NSF. And I'm perfect timing. Thanks. Thank you. About CC, okay.
You mentioned
Yeah.
Could you say something about how to graph class?
How to this graph clustering? There are some parameters that we need to do, right? Yeah.
How to make the graph and then
How do we make the graph? Those dating cluster is for graph data. It can be applied to graph data. Yeah. There are some parameters you need to adjust.
no.
Mm-hmm. Yeah, yeah, yeah. And so that's so after we normalize that, we do the clustering. So the age weight is just a normalized count.
That was very good talk. I wanted to ask if you, I know most of your sort of experiments for validating were based on computational simulations of.
Yeah.
I was just wondering. Yeah, go ahead.
I mean, these are based on the real data, and then after we get the cluster result, we use the like a check M for the micro for the bacteria, and then we just see how many of them near complete those kind of stuff.
Right. And do you have sort of some results on the consistency? So do you see, for example, different viruses being joined together in the context falsely?
Okay, so that's what I asked Yushan to check, because in particular for the virus, you see the assumption, Jed and I at first don't feel that comfortable, and whether there are some two viruses that they affect the same host, but then they might link to the same host contain. And so what we ask him, you need to make sure that we are not mixing all those viruses affect the same host together. And so he did check carefully, and then yeah, there are some threshold issues. So if they are linked to the same host can take multiple times, not just one time. And if it's just one time, it's probably not good. But it's after you threshold on something, and they, I mean, he showed that. Oh yeah, that's why he does the last step. He checked all the proteins in that viral contain, try to see are they from the same family. If they most of them from the same family, then you are not contaminated by different viruses. Yeah, that's a very good question. And Jed and I try to push him very, very hard, because we are not that comfortable at first about those the host mediated graph.
Thanks. I like the talk very much. In one slide, you mentioned about application of the detection of horizontal gene transfer.
We didn't. I did not get.
Do you say something about that?
That's the. I mean, that's. I mean, you need to have different samples and then try to see if the viruses link the virus host, if they change as your condition, like you have some antimicrobial resistance genes get into it. Yeah, we did not look at too much into it, but that's the more on the application side, viral hosting.
One question. So by sequencing the gene, I think can you just identify, for example, what virus like a specific
Effects.
host is that identified?
Yeah, I mean, you can identify some of
Okay, and uh, do you have any quantitative study? For example, a virus can prefer a host over another. So you can, for example, you can define a parameter like intensity or.
them. I mean, that's the kind of things that we might not particularly this one, but you can use that one to like a machine learning kind of thing, just purely based on sequence
Yeah,
data.
yeah, yeah.
That's more kind of like the talk from last year. And you
Oh,
might be
okay.
able to,
Oh,
you can, I mean, for those of you who are interested in this topic, and then you can look at my last year's talks through YouTube or Bilibili. Idea.
Okay. Thank you. Okay, let's thank the speaker again for this very interesting talk.
Okay, thanks everybody for participating.